All right, here's our 11th video. We've done 11 videos in living environment regions. We're talking about tRNA, which is transfer RNA. So let's get our pen ready. Maybe we'll stick with red. We'll stick with red. It's the transfer RNA. Last video we talked about mRNA. We also mentioned tRNA, and we talked about protein synthesis, right? And we're continuing that translation now because we talked about transcription, then we talked about translation, but now we're talking about tRNA. So I'm sure you know that by now, right? And in the last video, we talked about these types of RNA polymerase. There's a type 1, there's a type 2. Oh, there's a type 3 here. Let's get our pen going. I put 2 twice. Let's fix that. Type 3. Right. And we said the type 1 was for rRNA, which we'll talk about soon. Type 2 was, we talked about in the last video, type 2 was the mRNA, the messenger RNA. And we have the type 3, here's T for type 3, which is the tRNA. So the RNA polymerase type 3 will make the tRNA in the nucleus. So tRNA is made in the nucleus. And you should know where the nucleus is. We're eukaryotic organisms, so we have, guess what? We have a nucleus. So it's made in the nucleus. Nucleus, right. And again, we said in the last video, the RNA is actually not made in the nucleus. Where do you think it's made? Do you remember where that's made? Right, that's made in the nucleolus. The nucleolus, which is inside the nucleus. All right, so let's continue from there. So after tRNA is made, let's just sum this up very briefly, and then I'll go over it slowly. After tRNA is made in the nucleus, so let's say that's the nucleus, it leaves the nucleus, and it's in search of used to be a TV show in the 70s. It's in search of this, and I'm going to write this long. Well, first of all, it's in search of amino acids. That's what it wants. In search of amino acids. When it leaves the nucleus, it's uncharged. That's a word to know. It's uncharged. It wants to be charged in order to be functional. So it's in search of amino acids. Once it gets its amino acid, it becomes charged. So now, where does it get its amino acid? Now, there's amino acids in the cytoplasm, and we get that primarily from food. The blood has a great supply of amino acids that they can give to cells for this reason. So it's in search of amino acids. Once it gets the amino acid, it is charged. Now, here's the alert. I'm going to write something. Let's clear this out. Wait a couple of seconds. 1, 2, ready to clear. We'll clear it out. It's going to look for amino acids, and let me write this word down, and then we'll say it together. All right. So we'll start it slowly. Amino, that's the first one. Acyl, amino acyl. Say that. Amino acyl. I'll put a little hyphen, tRNA. So let's see, read it so far. Amino acyl, tRNA. And then synthetase. So I'll write that down. Synthetase. So you say it a few times and your teacher will be impressed, right? Amino acyl tRNA synthetase. It is a molecule that the tRNA is going to look for so it can get its charge. It can get its amino acid. So this is very oversimplified. So let's say this is the tRNA, right? It's the tRNA, kind of looks something like this. Very oversimplified here. And I'll go over this again in a minute. So it's going to look for this molecule. It's going to dock on this molecule, which is the amino acyl tRNA synthetase, and it's going to get its amino acid. And then it can go to the ribosome to finish translation. We talked about that with the mRNA in the last video. All right, and here's another oversimplification of the tRNA molecule. So, and what's missing here is its 3' arm, right? And that's after it gets its amino acid. You can see it has this anticodon here. It's going to go into the ribosome. And I showed you this picture in the last video, but let's go back for a minute. All right, so over here, it's a little longer. It's this 3' end. So this whole thing, as I'm drawing this red line, this whole thing in the tRNA, it's all made up of nucleotide bases. And we talked about nucleotide bases. Remember, there's A and G and T and C, but T, thymine, does not leave the nucleus, so we replaced it with uracil. So they're all connected all over this area. So the T-arm, and we can think of the T-arm as the transfer arm, even though it's not called that. That's the transfer arm. That's the arm that's actually going to go into, or tether, we should say. It's going to tether or lock onto the ribosome. And I guess you can remember T for transfer. The D arm, some people use as the docking arm, even though it's not officially called the docking arm. So just use it for your own mnemonic device. The docking arm is going to dock, and I'm going to write it again. See if you remember. See if you can say it as I write it. Amino acyl, T, and what do you think the last word is? Synthetase. That's it. See if I fit it in here. Can I fit it? Oh, good enough. So this arm, the D arm, is going to dock into this molecule, which I'll show you in a minute. I briefly showed you a rough drawing of it. And over here is the anticodon. The anticodon, that's the one that's going to take the codon from the mRNA and create the protein. All right, so look at that for like 2 seconds. Now, this is also called up here. Let's change the, put a little arrow here. This is also called the acceptor arm. What do you think it's going to accept? The acceptor arm. The acceptor arm accepts, fix that E. There we go. The acceptor arm accepts amino acids. Well, just one, an amino acid. It's going to accept its amino acid. So let's look at a better picture. All right. So, and let's get our pen. Let's do violet or purple, whatever you want to call it. So here it is. Amino acyl tRNA synthetase is this molecule where the tRNA is going to dock. It's going to attach itself. It's going to become charged, and it's going to get an amino acid connected to it. So this amino acid, right, is going to be attached to it. And remember this, we talked about this when we talked about how cells get energy, ATP. Does anyone remember how that is? I'll write it over here, ATP. It's adenosine triphosphate. Let's write that out. Even though you're just probably going to hear it as ATP, right? So adenosine triphosphate, which gives the cell energy, right? And it's going to give the tRNA energy, right? It's an energy currency, you can say. So again, the tRNA is going to dock in the amino acyl tRNA synthetase. It's going to be charged and get an amino acid. And here it is on the bottom. So let's change the color. There we go. So here it is. It's all ready to go. It's charged. It has its amino acid. And where is it going? It's going to the ribosome. And let's look at that again. So here we go, right? So it has its amino acid. It's charged. It has its codon. It's going to take and this is it has its anticodon. Let's get a little pen. Fix the pen. Right? It has its anticodon. So let's look at an example of this. So let's say it has alanine, right? Alanine here. It's amino acid alanine. And alanine is GCA. So on the tRNA, it will have GCA. And it's going to look for a codon that matches that nucleotide. So that would be CGU, right? So let's say it's here, CGU. And it's going to attach, and out will pop alanine. And it'll do this for over and over until it creates this peptide chain, which are short chain amino acids. So that's basically what happens with tRNA. Now, there's another word for these. Let's go back 1 second. Okay, let me just, okay, I'm going to draw. Get the pen going first. Go back. Let's get the pen going. All right. So if I draw this simplification again, we have this boom, 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 and so forth. All right. You get the idea. All right. So there is an ACC, we should know. ACC, or CCA actually. You read it backwards. CCA, it reads ACC on top. The ACC is where it can carry the amino acid. And the way people remember it remember, read it from the bottom up. CCA. And this is an easy way to remember it. Can carry, what do you think the A is? Amino acid. Not acids, amino acids. Only going to carry one. When it docks in the amino acid tRNA synthetase, that molecule, when it docks in there, it's going to dock based on one individual amino acid. So if it doesn't have a code for a certain amino acid, it will be rejected. So it comes in, right? Let's just say something like this. Here's where the amino acid is waiting, let's say, and it comes in, it docks, right? It's docking, boom, boom, boom. Right? Has its ATP here. That's going to help it get charged. If it doesn't have the right code for this amino acid, it'll be rejected. It'll leave, right? The amino acid tRNA synthetase will say, no, we don't want you, go away. But if it matches, it will grab the ATP, it'll grab the amino acid, and head to the ribosome. All right. So now we'll carry on to video 12, which will go into more of some biological processes.